I'm Sumit Karai, and what we have on the screen right now are a bunch of ants. And these ants, let's say, are in the wild, and as they're in the wild, they need to survive, and survival needs resources. Let's say something like food. So there's food out there in the wild, and ants will need to detect this food, and then they need to reach the food, collect that food or any kind of resource that's necessary for survival, and get that resource back to their home or their colony, right? So this was pretty easy, but this was a very idealistic situation which does not occur in nature, or even if it does very rarely. Nature is difficult, and the path from the colony to the food can be highly complex and difficult to navigate. Not only that, nature or the environment surrounding an ant's colony can be highly dynamic and can change very quickly at a moment's notice. So in this complex and dynamic situation, an ant needs to gather resources. How does it do that? So it has to reach the resource, and it has to understand how the path changes, and also it has to remember that the path has changed, and then so that when it comes back to gather the resource, it can follow the same path. Otherwise the resource gathering will not be efficient. But how does that happen? How can such a small organism do that? So even if they are so small, they have some basic neural processing built inside them, and this neural processing allows them to go through the environment and learn about their environment, and this learning is then stored inside their small processing units in the form of memory. So this is how something like an ant navigates the environment. But then memory is something that is finite, because storing memory requires resources, and obviously an animal, if it has to survive in the wild, it has to manage resources in this complex and dynamic environment. So with learning and memory, there also comes something called extinction, where an ant will slowly forget what it has learned. But the question is, all of these concepts are very abstract, and how do we experimentally, quantitatively understand these concepts? And that's where we borrow something from the field of psychology called conditioning. Now what conditioning is all about is, it's actually very basic and very simple, and one of the easier ways to understand the process of memory and learning. So how does it work? So you have an organism, and we want the organism to perform a certain behavior, certain favorable behavior, and when it performs that behavior, like here, it goes and grabs the letter G, we give it a reward, right? And as it gets a reward, which is something favorable to the organism, it will repeat that certain behavior, and that's what conditioning is all about. So what I'm trying to understand in the experiment that I attempted for the ecology project was, can these ants be conditioned, and if they can be conditioned, how long do they hold this conditioned memory inside their tiny little processing units, and also if this thing is possible at all. So, and also how much time does it all take? So let's go and explore this in further detail. So let's take a look at the experiments that I did to understand the memory and learning in ants by performing conditioning. So what you see in front of your screen is a Y-shaped structure. Let's just call it the Y structure. This can be made up of anything. So what this structure is supposed to do is, it's supposed to let an ant enter it and then as it goes, there's a fork towards in the road, and it has to then decide if it has to go towards the left or the right. So it makes the decision, and then it goes towards a certain direction, whichever direction has been decided by the ant. But the question is, what should the structure be made up of? So in my initial attempts, I tried to make the structure out of flat PVC pipes, and I put that entire thing inside a water trough, and then I tried to see if the ant would navigate the structure, but to my dismay, the ant would try to escape as soon as I kept it inside this structure. And even though I thought that water would deter the ant, but the ant jumped right in the water and swam away. So I had to ensure that these ants remain inside the structure and do not get away. And for that, I thought, okay, let's just make a structure and put a layer of cellophane over it, transparent cellophane attached by transparent adhesive tape, so that once the ant is inside, it does not leave. Now that I had taken a cellophane paper and covered my Y structure with it, I had ensured that the ants, if they enter the Y structure once, they would not be able to escape it midway. They will have to complete the entire maze, and only after that would they be able to get away. And so I breathed a sigh of relief, because now my experiment could be done easily. So I just started putting the ants inside this cellophane paper, not the cellophane paper, but Y structure covered in cellophane paper, and this is how it looked like. So what I tried to do was catch individual ants from the ant housing and put it inside the maze, so that they could traverse the maze and do whatever they wanted to do. And here you can see that this is all being done inside the ant lab of IISA Kolkata, whom I'm very grateful for. Not only Dr. Sumana, who is the primary instructor of the primary investigator of the ant lab, but also the masters and PhD students who helped me a lot. But as I noticed while I was doing the experiment, was once the ant has been handled using those certain tongs, they sort of felt stressed out, and I thought that stress might add to it might affect my experiment, and that's why what I thought was, why not put the entire colony in front of the maze, so that the ants can go and enter into the maze whenever they wish. So I just took the entire ant nest, which is kept inside a red a petri dish, which is covered by red cellophane paper. And I took the entire ant nest and put it inside this box, which was attached to my Y maze, and I also attached a gate so that only one of the ants could be allowed at once, so that they can go and my experiment could be done, which I'll describe later. But this is how it looked like in real life, not as geometric or accurate, but it did the work. So this is how it looked like in real life. When the experiments were being done, I put a wire mesh over the rectangular portion where the nest is kept, so that the ants have no way to escape. And what you see is the Y maze or the Y construct connected to this certain apparatus, which allows only one ant at a time. So now let's quickly take a look at the 3D structure or 3D model of this entire apparatus. This is how it looks like. We see that this entire Y structure has these two arms, one of which is connected to this rectangular box, and the two arms are open and they are sealed at the end. And so this is the gate, which can be regulated to let only one ant in at a time, and this ant will now enter. And as you can see, this ant has entered, and now it has to make a choice, which it will. And after it has made a choice, it can go towards either direction. So now, the first thing that I wanted to work with was to remove any sort of directional bias. So what I wanted to see was if these ants have some preconceived notions about which direction to choose. So what I did for that, to remove directional bias, I simply took the naive ants which have not been tampered with and let them cross the maze, and I saw the proportion of ants that took a certain direction than the other, and I saw after doing the statistical analysis that there is no directional bias. So that was done, and you can read about it in detail in the report. And then comes the important part of training. So what happened in the training was that I had to somehow familiarize these ants to the maze and also make them choose a certain favorable behavior that I wanted them to learn. So I detached my Y structure from the entire apparatus, and this Y structure was kept inside the ant housing. So this is the ant housing, and this Y structure is kept inside the ant housing, and on one side of the Y structure is some sort of positive reinforcement, that is food, and all other sources of food are removed. Only that is the only source of food, and this is how it looks like in real life. And there is where the food is supposed to be kept, so that whenever the ants enter this maze or this construct, they not only get familiarized with it but also understand that there is food at one direction. And now this entire apparatus was kept inside the ant housing, and it was kept for 24 hours. And after 24 hours was the testing phase. So we just took the entire ant colony out and inside the box where the testing is supposed to be done, and then I saw or I recorded whatever they chose, if they chose to go towards the left direction or the right direction, and I noted down my observations. And this entire cycle of detaching the Y maze, keeping it inside the housing colony, and then testing it again after 24 hours was done 3 times. So one cycle consisted of all of that, keeping the colony inside keeping the structure inside the ant housing for 24 hours and then testing, and it all was done 3 times. And what were the results after the testing? It was seen after all that the proportion of the ants choosing a certain direction was not statistically significant to have any sort of bias development, which means that they did not develop the training, they did not get conditioned to choose a certain side, at least according to the experiments that I did. So the testing was done. So now coming to the conclusion, or what went wrong, in other words, well, the first problem that was there was the lack of time. I did the entire cycle of training and testing for 3 days, but in literature there are reports of achieving this in 6 days or 12 days. So that is something that has to be kept in mind for the next time the experiment is to be done, is that more time has to be given for the ants to get familiarized and get trained in the behavior that we are trying to get them trained in. But I did not have the luxury of time, because I spent a lot of time figuring out how to keep the ants at one place, and also to learn handling and solving the small problems that comes with making an experiment of your own. And the next problem could be that the food that was there as a positive reinforcement was not enough, and maybe there needs to be some more positive reinforcement, or maybe there also has to be some negative reinforcement or some sort of a punishment also, to make the understanding, not the understanding, but the learning of the behavior even more quick. So because there was no training, I could not perform an experiment on extinction. So even though my experiment does not provide a clear view on how the memory and learning inside an ant works, but future experiments might provide us with a more focused understanding. So the entire project was done by Sumit Gray, and I'm very thankful for our ecology instructors for giving us this wonderful project in which we not only did a lot of fun stuff but also understood how engaging it is to develop an experiment from scratch. Thank you.